Great. Thank you so much, Sarah, for the invite. I'm very excited to be here today to talk to you guys about our recent work entitled Identification of Cell Type-Specific Cis-Regulatory Elements in Alzheimer's Disease. This was recently published in Cell Genomics, and so if there's details that I leave out that you're super interested in, ask, or feel free to look up the publication. So the goal of this project is really to understand gene regulatory mechanisms in Alzheimer's disease. And as most of you are probably aware, there's been an explosion in single cell genomics recently, specifically in single cell RNA-seq. And a lot of work has been done in this area, specifically on Alzheimer's disease. You can just see from some of the publications, which I've tried to list here, this is probably not all of them, but a lot of work has gone into identifying differentially expressed genes in specific cell types that are relevant for Alzheimer's disease. A few of the examples are shown here. Now, in addition to this, decades of work have gone into understanding genetic contributions to Alzheimer's disease in the form of undertaking large-scale GWAS studies. And so to date, I believe there's about 80 risk loci that have been identified through numerous studies. And the goal of our project was really to kind of link these together and to understand how non-coding genetic variation influences the differential gene expression we've been seeing in these single cell RNA-seq studies, and try to really dig into, how are these different cell types being regulated, and how are the different genetic mutations affecting that cell type specific regulation. And so for our project, we went into this right at a great time when the 10x single cell multiomics kit just came out. We were just getting our samples and starting to design this experiment. And so we immediately jumped on board with that. And we've got 7 sporadic Alzheimer's cases and 8 controls. We got DLPFC from these individuals and performed the single cell multiomics. And single cell multiomics, for those that may not be familiar, is essentially you are isolating single nuclei through the 10x system. And from a single nuclei, you're profiling both ATAC-seq signal as well as gene expression, because both the DNA and RNA have a cell barcode that is unique to that individual cell. And so you can put the data back together without having to infer or first classify cell types and try to figure out whether this ATAC signal goes with the cell. You already know that information because of the barcode. So first, we identified a total of over 105,000 nuclei and sorted them into, or identified different cell types, as one does. And we found all of the cell types that one would expect within DLPFC, as shown here. And we've looked at gene expression of marker genes for each cell type, like alpha-4 for astrocytes and GAD2 for inhibitory neurons, as shown here. And we also looked at how well accessibility at these marker genes matched up with cell types. And you can see from the panel on the right that at the marker genes, you had the most accessibility in that specific cell type for which the marker gene identified. In addition, we identified subpopulations, and then we looked to see if there was uniform donor contribution within each cluster, mainly to make sure that one donor wasn't overwhelmingly contributing to a single subpopulation. And we found relatively even distribution across all of the cell types and subclusters that we identified. Not unexpectedly, we also saw high variability in the cell type proportions among donors. So for any individual donor, we would see a variable proportion of different cell types captured. Generally, we found that most of the cells from any given donor were oligodendrocytes, and this is consistent with the previous literature in the single cell RNA-seq field as well. The second most abundant cell type were excitatory neurons. The first analysis that we did was to look for cell type specific differential gene expression in Alzheimer's disease. So within each cell type, we did a differential gene expression analysis, and shown on the left is just a heat map of the up and down regulated differentially expressed genes that we found in each cell type. We identified 911 of these, and you can see here that we see some of the genes that have already been associated with Alzheimer's disease in these different GWAS studies, such as tau, CLU, BIN1, ApoE, etc. Here's on the right is an example of several of these differentially expressed genes, SAMD4A in astrocytes, PTPRG in microglia, and then so on and so forth. PTPRG in microglia is important because it was shown to be upregulated in Alzheimer's disease in all of the single cell RNA-seq studies that we examined. So we're seeing similar things that have been shown previously in the RNA-seq or the gene expression profiles. When we do a Go analysis of these differentially expressed genes in each cell type, and we split them into upregulated and downregulated DEGs, we see pathways that, again, one would expect in Alzheimer's disease and things that, again, have been reported before. So in microglia, for instance, we're seeing upregulation of PDGFR signaling in response to LPA and inflammatory type pathways. In neurons, for example, we see downregulation of things related to tau protein kinase activity, and as well as things affecting calcium homeostasis. And again, these are consistent with findings previously reported in the field. So we have good confidence that the gene expression profiles that we're measuring are of good quality. So now getting to the power of multiomics. So having both ATAC and gene expression really allows us to identify these cis-regulatory elements that we're really interested in. And to do so, we're going to calculate correlations between accessibility and gene expression. Now, there's several ways in which one can do this. And when you don't have paired and matched RNA-seq and ATAC data, you have to be very conservative in your correlation estimates, because you're not saying that this ATAC peak was open in the cell that had this expression profile. But because we have multi-omics, we have a lot more confidence in our data and a lot more confidence in the correlations that we can make. So what we do here, and just to familiarize you with some of the terminology we're going to use, is when we have an ATAC-seq peak, we're going to call it a linked peak because it's linked to a target gene's expression, and that will be called a linked gene. The link itself is not a physical link like you would see in Hi-C or other contact-based assays. This is simply the correlation itself. So when I say link, I mean correlation. And the way these are calculated is kind of illustrated in a cartoon here, where in an Alzheimer's cell, for instance, we have this candidate cis-regulatory element that's accessible, and we have a high level of gene expression. Whereas in a control situation, that region is not accessible, it's closed, and the gene's expression is lower. So this would indicate a positive correlation between this region and this gene in AD, specifically in AD samples. So we can have multiple links per peak. So a single cis-regulatory element can be linked to multiple genes, and on average, we get about 2 links per peak. Conversely, you can have multiple links per gene. So a single gene can be regulated by more than one cis-regulatory element. And our median for this is 14, although we have some on both ends of the spectrum, as far as many genes have 1 or 2, and then a few have a lot more. In order to identify these links, we first recalled our ATAC peaks in each cell type so that we wouldn't lose any of that cell type specific signal from less abundant cell types. A key feature of our analysis was that we analyzed AD and control samples separately. And this allows us to identify disease-specific and control-specific linkages, so things that are only correlated in a disease state. And then, of course, we filter links based on significance and correlation values. So across all cell types and all samples, we identified 318,000 links. Here's just an upset plot kind of how these links are distributed across control and AD samples. You see, for the most part, they are similar numbers, so we didn't skew in any one direction significantly for either control or AD. Shaded here in gray are the cell type specific links. And you can see that the most were identified in excitatory neurons. And again, this was a very abundant cell type across the data set. And there's a lot of expression diversity within subpopulations of excitatory neurons, which is part of why we're able to identify more correlations, because the range of expression variability is quite high. We can also see here, I grouped excitatory and inhibitory neurons together to also show just a neuronal set of linkages here that I'll also term cell type specific quite often. The next most abundant category here is the ones that were shared across all cell types. And these are going to be linkages probably for more housekeeping type genes or genes that are just expressed in general cellular functions. So again, to see, this is a lot of linkages. So what evidence do we have that any of these are actually real and are actually capturing real regulatory elements? And so one of the first things we did was to overlap these with ENCODE CREs. So these are categories of the genome's basically been partitioned based on many chromatin modifications and DNA hypersensitivity assays and whatnot to classify them as either, we'll focus mainly on distal enhancer-like sequences or proximal enhancer-like sequences. And then red here is promoter-like sequences. And so you can see that most of our links are overlapping these enhancer-like sequences, either proximal or distal. In addition, we examined previously published data sets that had looked at H3K27 in human brain samples that were looking at sorted populations. So from Knott et al., we got the astrocytes, neurons, microglia, et cetera. And then from Kozlenkov et al. From Stella Drasheva's group, we looked at the GABA glue K27 acetyl data. And we broke down our linked peaks into two categories, either shared, so it's an astrocyte peak, but it may also be a peak in another cell type, and looked at the proportion that overlapped K27 acetyl signal from that same cell type. And you can see it's usually between 30 and 50% across the cell types. But when we look at specific peaks, so these are linked peaks that were only found in astrocytes, we generally see a higher proportion in some of the glial subtypes, particularly. We think that some of the overlap within the neuronal populations, because neuronal the epigenome is very experience dependent, it may be because these weren't the exact same samples, you're seeing less of an overlap there. So here we're looking at our links in a little bit different way by looking at the linked genes themselves. So here on the y-axis are all of the linked genes. And then we're looking at, in this first box here, the most correlated linked peak and its accessibility score. And then we're looking at the expression of that linked gene over here in the greenish boxes. And this is split up by either Alzheimer's specific links here at the top, common links, which of course is the biggest group in the middle, and control specific links here. And what you can see is that for the common linkages, we're plotting the correlation values here. So the higher correlation values seem to be found in common, but there's a lot more data supporting that. So that's not unexpected. But we do see that there are reasonably high correlation values for both the AD and control as well. And you can see that largely when it's accessible, you see high expression. So the majority of our linkages are positive correlations. But again, I told you the power of our analysis is that we're really able to identify these disease-specific links. And so when we look at those, we've identified 40,000 links that were only correlated in Alzheimer's samples. And similarly, we have 73 in control, and of course, the majority were common. When we look at the distribution of cell types that these links were identified in, we find that there was a higher proportion of microglia linkages that were AD specific compared to any other categories. And again, this is evidence supporting the fact that we're really picking up information relevant to Alzheimer's disease because we know how important microglia are to Alzheimer's disease biology. But let's get back to the initial question that we really asked, which was how does non-coding genetic variation influence this differential expression that now we've cataloged? So the first way to address this is to ask if there's disease-associated genetic variation in the linked peaks that we've identified. And to do this, we performed LD score regression. And this is a similar analysis to many people have done, where you're basically partitioning heritability of a trait across the genome and asking how much of this variability does your data type explain, in this case, our linked peaks. So what we've got is we've split our linked peaks into several categories, all of our linked peaks for that cell type, common links, AD-specific links, and then control-specific links. So this is these colors here, and we've done this across all cell types. So when we focus on Alzheimer's disease, not unexpectedly, we see the most significant enrichment, and those are indicated by numbers in these boxes here, are found specifically in microglia as opposed to any other cell type. In addition, we find this enrichment for all our links, common links, and AD-specific links, but we find no enrichment for our control links, again, supporting the fact that we have a very specific signal that's really relevant to Alzheimer's disease. And again, just pointing out that it's really only in microglia, and this is consistent with previous literature as well. Now, as controls, we looked at other brain-related disorders and immune disorders, as well as other, height and BMI. And for other brain-related disorders, we see things pop up in neurons for, schizophrenia and bipolar disorder. And then, of course, nothing really for immune and then the control samples, as one would expect. So then we would ask, well, do our differentially expressed genes have disease-specific links? So here I'm just showing a quick percent of DEGs that were either decreased in expression in AD or increased in expression in AD, and asking how many of those genes have a control-specific or AD-specific link. So when you look at things that were decreased in expression in AD, they tend to have more control-specific links. Conversely, when they're increased in expression in AD, they tend to have more AD-specific links, suggesting that the link, when genes are being overexpressed or increased expression in AD, they're either picking up new cis-regulatory elements or they are losing control-specific regulatory elements. To the right's an example for a DEG that was downregulated in all cell types in Alzheimer's disease. This is cancel1. Now it's found in the tau locus. You can see that right here. And what we've got plotted are the ATAC-seq signals for the AD samples or the control samples, specifically in excitatory neurons. And you can see in the region that I've highlighted that there's a decrease in accessibility in Alzheimer's disease compared to control, and that this is a link that's overlapping a previously identified SNP here from this recent EQTL study here, regulating cancel expression. And so here's just an example of a link that we've identified, regulating a gene important for Alzheimer's disease biology.
Lindsay, can I ask a quick question?
Of course.
So do you generally see this overlap between differential expression and downregulation or upregulation of the linked regulatory element in terms of the ATAC signal?
So I will say for single cell attack, specifically in postmortem brain, the ATAC signal can be messy and difficult to look at. So there's a few examples where this is very, very clear, but a lot of the examples that we see are a little more muddy. And so we're really relying on our computational pipelines to pick out those regions that are important. And so all of this goes to say that anything that we're identifying in these single cell studies has to be followed up on and validated. And really what we're trying to do is capture kind of the realm of possibilities that make the most sense for us to follow up on later. And so kind of what I'm doing as I go through the talk is really just building up the evidence that we've identified 300,000 links. They are clearly not all real. How are we going to go about narrowing this down to what we believe is really important? And overlapping with things like these EQTLs is one way to do that. And then we'll get into some other ways that we've done that as well. Overlapping with K27 acetyl, like I mentioned previously, is another way.
Right. Yeah, that makes sense. And would you say then that you have better power to detect these disease-specific links than power to detect differences in ATAC signal between cases?
I think so. I think because you're combining the two, you're really lending power to both assays. And so, and again, having this multiomics kit gives me a lot more confidence than I would have if I was just trying to first establish like co-accessibility with a promoter and then going in and looking at expression, because previous studies that have done that, they've got almost tenfold less links than what we're identifying. And so, you know, they have just a different set. And so I think there's different strategies to go about it. And we were trying to take a broad strategy and then narrow down in other ways, just so that we're not missing things that could be really important.
Great, thanks.
So again, we're back to this question of how are we going to narrow this down? And so to do this and to really understand which transcription factors might be contributing to this the CRE activity, we devised a strategy where we're looking at these trios. And about the same time, I'll go through this, but then about at the same time, I just want to point out that a method called tripod was published as we were submitting this paper, and it's really built on the same idea. So if you want an in-depth explanation of this, as well as some extended functionality, I recommend that you go check out this paper. But it's built on a simple principle that I'm going to go through now. So we've already established these linkages between ATAC peak accessibility and gene expression here. So that's again illustrated here in one. We're identifying linkages. Each linked peak for a transcription factor motif, and then look to see if the accessibility of that peak was correlated, right? And so leg of our triangle and say, is transcription factor now correlated with true? Then we consider that. Does that make sense? so looking at the entire data set, we identified over 60,000 of these trios. Importantly, 66% of our differentially expressed genes participated in a trio in the same cell type in which it was differentially expressed. When we look at all of our trios and we ask, well, which are the transcription factors that seem to be playing important roles? What jumped out to us was MEF2C. It's involved in 5% of all the trios identified. And here, what I'm plotting is in each cell type, even if it didn't have a trio in it, what I'm pointing out is the expression of the linked gene for all of the MEF2C trios across all cell types. And what you can basically see is that you get different linked genes in each cell type for these trios, right? And so for excitatory neurons, when we do a GO analysis here, just in neurons in general, we see MEF2C regulating things like synaptic transmission, potassium ion transport, cation channel activity, neuronal type things, which one would expect. When we look in microglia, however, we're looking at things that are more related to immune response, pattern recognition receptors, cytokine production, et cetera. So even though it's the same transcription factor, in different cell types, it's regulating very different transcriptional programs, which is not a surprise to anybody on this call. But it was an important thing to see in our trios and validating, you know, that we're picking up things that are real. And this is just showing MEF2C expression across all of these different cell types. Back to looking at kind of total trios, which I skipped this part by accident. When we look at kind of where these peaks are that are involved in trios, most of them tend to be intronic or distal intergenic regions here. And so they're in these areas of enhancer, where enhancers can tend to be. So again, getting back to the power of our analysis, we can still split these up and look at AD and control specific trios. So now we're getting down to numbers that are reasonable to go through and test. So we identified 2,700 specific trios, AD or control specific trios, and it's about evenly split between the two. We find 96 DEGs in AD specific trios and 89 in control specific trios. And then there are 30 DEGs that are found in both. And so when we start to plot this, what we're looking at here in these heat maps are for the AD samples or the control samples in a given cell type, we're plotting the peak TF correlation. So remember, that was the second leg of our triangle here, peak TF correlation, and then our gene TF correlation, which was that third leg of our triangle for each of these AD or control specific trios. So the bottom is our AD specific, and the top is our control specific. And you can see these correlations are higher in control or AD, as shown here. Then plotted on the right are just a color-coded plot of which TFs are anchoring these trios. And so in control-specific trios in microglia, we see a lot of red, and that corresponds to NR4A2 or NR1. This is a protective, anti-inflammatory transcription factor that binds NF-kappa B targets and recruits the co-rest repressor. In AD-specific trios, we found the most predominant color to be blue, which corresponds to MAFB. Also, MAFB also promotes homeostasis and anti-inflammatory phenotype in microglia. And so what we're thinking is happening potentially is that there's a switch from the way you're kind of dealing with inflammation in control versus AD samples. And of course, this would need to be followed up on a little bit more. In neurons, when we look at the same kind of heat map here, and this is trios that were found in inhibitory or excitatory neurons, we see that in the control scenario, we see a lot of purple, which corresponds to KLF10. And in AD-specific trios, we found the majority of trios involve ZEB1. And this was really interesting because ZEB1 hadn't been previously implicated in neurodegeneration or Alzheimer's disease. It's been associated with EMT transition and glioblastomas. And so we wanted to follow up on this a little bit more to figure out if this was real and what it could be doing since it's playing such an outsized role. So here is just an example of kind of the way we went about following up on this. So this is an example ZEB1 trio in excitatory neurons. Here I'm plotting the accessibility in microglia, which it's not accessible, in inhibitory neurons and in excitatory neurons. To validate these, we performed ChIP-seq of ZEB1 in DLPFC tissue from our 2 control donors. And we found a ZEB1 chip peak overlapping this ZEB1 trio here. So here is our link in green, and these green boxes are ZEB1 motifs. So we've got a region of accessibility and excitatory neurons that corresponds to ZEB1 signal in ChIP-seq overlapping a ZEB1 binding site. And it also overlaps a region of neuronal K27 acetylation from not at all. And this is found within the GABRA5 gene. We can see here, this is the ZEB1 motif. It is it's a good motif. It's not just a bunch of G's and C's, which tends to be information poor. It's a strong motif. ZEB1's expressed in these cell types, but you can see that GABRA5 expression is predominantly in excitatory neurons. So this all holds up, and it all confirms the ZEB1 trio here. And so you could, of course, go through many of the trios and do the same kind of thing. So as I mentioned previously, one of those important things that we have to do now that we have all these linkages is to really go about validating them and say, okay, which of these do we really think are functional? One of the ways that we can do this is using previously published functional assays. Some of the assays that we looked for in the literature were MPRAs. So these are massively parallel reporter assays that were performed in NPCs, neuronal precursor cells, embryonic stem cells, and then other cell types that this data exists in. And we found significant enrichment in the active MPRAs elements for each of these. So again, just an MPRA is you basically take a piece of the genome, put it into a reporter assay, and ask, can it stimulate activity? And so of the active elements, again, we found an enrichment of those overlapping our links. We can also use EQTL data sets that I mentioned previously, and we pulled 2 from cortical tissue from the recent 2022 study, as well as from GTEx. And again, we see a significant enrichment in our data set. Finally, we use UN high-C data from Hugh et al. 2021. And again, we see a significant enrichment in high-C contacts in that match, not the region being linked to the same gene that we're detecting a link. So everything, there's just a lot of positive data supporting our findings. But of course, we really need to go in and experimentally validate these things. And so we are taking 2 strategies in which to do this. And we're validating only common or control specific neuronal CREs at this particular point, because we would need an AD cell model, and we just haven't gotten that up and going yet, but it's on the docket. So for now, we're sticking to these.
Putting everything together.
Yeah.
Is there a question?
No, sorry. I think someone accidentally unmuted themselves.
Oh, okay. No problem. So one of the first strategies is using luciferase assays, in which we insert one of our elements into a luciferase reporter. We transfect that into cells and then look for luciferase activity. And so that data, using that strategy, we have validated several of our CREs that were linked to these genes shown here on the bottom. So what we're looking at is the luciferase activity compared to the empty vector control. And you can see that these regions all stimulated activity here. And several of these, of course, SNCA here, PHF24, ADAM11, ADAM-TS1 are all linked to Alzheimer's disease. What these luciferase assays also give us the ability to do is to introduce mutations into these elements and see if that mutation influences the ability of that element to promote activity. And so there's one example for JPH3. So we had a region, a CRE that was linked to this gene that promoted activity. And then there was a SNP in this region that, when introduced into the element, abolished the ability of it to stimulate activity. So this is one example where we can test these individual SNPs. And JPH3 is actually has an expanded trinucleotide repeat in it associated with a Huntington-like a disease. And so we thought that was interesting.
That's quite interesting. And do you know, I mean, that would be like the full picture, I guess, but do you know if there's a specific transcription factor involved in that? And is it disrupting its binding?
Yeah, so those are some of the next steps. For that particular one, we didn't have, because a lot of times what we'll have is several motifs that kind of stack up around there. And so it's difficult to parse. So that particular mutation did not have a predicted disruptive effect on a motif. But there's also a lot of variability when you start looking at cell types. If you would call de novo motifs in different cell types, the motif can change slightly.
Yeah.
And so I think we don't really have a complete picture of transcription factor motifs across all the cell types that we're looking at right now.
Oh, yeah, it's definitely true, I think.
So another region that we focused on was the APP locus. Obviously, this is really important, the amyloid precursor protein. And so we wanted to drill down and find any new structure. And so shown here on the right, whoops, let me get my mouse in the right spot. Shown here on the right are the accessibility tracks in excitatory neurons, inhibitory neurons, oligodendrocytes, and OPCs. And the links here are all the links that we tested for CRE activity in our luciferase assay that are linked to APP. Most of these were common, so they were found to be correlated both in AD and control samples, and there's one in blue here that was control specific. When we performed our luciferase assay, we validated 3 regions shown here with asterisks and highlighted here in gray that were displayed the ability to stimulate luciferase activity. Interestingly, there's one negatively correlated link here that when we put it into our luciferase assay actually inhibited luciferase activity. So this assay isn't actually designed to detect repressor activity. So this is a region that we really want to follow up on because it agrees with both the correlation and the luciferase assay. So it's an interesting region that we think there's a little bit more study. So now we take another tack where we're going to start doing our CRISPR interference assays. And so to do these assays, we're using a differentiated iPSC culture model. This is the Neurogen N2 cell line where you differentiate into excitatory neurons. And to perform a CRISPR-I experiment, we're going to synthesize guide RNAs to our cis-regulatory elements, clone them into a vector that's got this dCas9 crab domain. So this is going to recruit repressors to that region and prevent it from being able to stimulate activity. And we introduce this vector into iPSCs, differentiate them, and then sort out GFP positive cells that got the vector, and then perform single cell RNA-seq on those. Sorry, I think my mouse is on the wrong screen. There we go. So when we do that, we did a pilot assay first where we're using 130 guides targeting about 60 regions plus controls. And we captured over 30,000 differentiated neurons. We analyzed those targets that had at least 100 cells per guide RNA to give us the power to detect differential expression. And as controls, we're using guide RNAs targeted to the AVS1 locus. So it's a safe harbor locus. As we found, targeting to GFP caused some strange results. And I think just because if you don't target the dCas9 to anything in the genome, it starts to be a little wonky. So to start, we look at some of the promoter guide RNAs that we used. So we designed guide RNAs to the BIN1 promoter and ATP2B4 promoter. And as you can see, when we target them, we see that they are significantly reduced in expression. We found this, you can see this again here in the box plots, where we're looking here at BIN1 expression and the control scenario is here. And then when we have our promoter guide RNA expressions abolished. And then similarly for the ATP2B4 expression in our control with our control guides versus 2 of our promoter guides expressions abolished. So we can also do this when we target our CREs, which is of course the whole point. And so when we look at 2 cis-regulatory elements predicted for SNATAC-seq, we see that we do reduce SNATAC-seq expression for both of these regions. But you will notice that the degree is a lot less than it was for our promoter guide RNAs. And this is of course to be expected because genes can be regulated by multiple CREs and there can be additive effects. And so, but you will see, at least in this case, it is the most significantly downregulated gene. And here it is as well for both of these CREs. And this is where the cis-regulatory elements are located, and they overlap 2 different SNPs that have been associated with ADHD. One of these overlaps a motif for both SOX21 and LIN54. And you can see it's predicted to disrupt this binding site in this particular instance here. And they're over 100 kb away from the promoter, closer to 200, I believe. And so we're really able to start picking up these distal cis-regulatory elements. And this is just one example. We're scaling this up to do a larger scale experiment now where we're testing a lot more elements. So another way that we can perform these CRISPR interference experiments is a much more low throughput way. And this has been done kind of a one at a time strategy where we clone in a single guide RNA and we differentiate iPSCs using a different strategy for this assay, using the Barty et al. Protocol from 2015, using brain fizz media, where we differentiate and get a mixed culture of astrocytes and excitatory and inhibitory neurons together. In this way, we can kind of see general effects, but we could also sort the cells and look for cell type specific effects as well. But we're basically cloning into the same type of vector. And again, we'll inhibit the cis-regulatory element and measure effects on gene expression, either with RNA-seq and or qPCR. And we've done this in a targeted way. Bree Rogers was the first author on this paper, focusing on the MAPT the gene, so tau. And we identified several new cis-regulatory elements for tau, as shown here. And if you're interested to learn more about that, I direct you to this paper, which was just recently put up on bioRxiv. But again, this is using a lot of our linkages were used in this, along with several other nomination methods. But it seems like our linkages tended to be positive in this assay more frequently than other methods. But we haven't done a large scale analysis to put any stats on that, but it's very promising. So with that, I'd really like to thank the two first authors for the single cell paper. Ashley Anderson was really amazing in doing a lot of the analysis for this assay or for this study, and Bree did all of the functional validation follow-up. And so these two were huge contributors to this effort. And of course, I want to thank our funding mechanisms and the NIH Neurobiobank and the donors and their families, without which, of course, we couldn't do any of this research. The Myers Lab has been great, and then Nick Cochran's been a collaborator on this project as well. And with that, I'd be happy to take any additional questions.